Today we shall talk about virus. Virus. Virus doesn't come under any classification that we have done till now. The 5 kingdom classification, they do not include virus. Virus is something special, which is studied just by itself. So let us see what viruses are. Virus, the meaning is poison. And virus was discovered by a Russian botanist named Dmitry Ivanovsky in 1892 when he was working in tobacco mosaic disease. He was trying to find out the cause of tobacco mosaic disease when he by chance came across virus. He was filtering for bacteria and the extract that he got after bacterial filtration, he used that again to inoculate for the disease, and the disease showed. After bacterial filtration also disease showed in tobacco. So he started wondering what is the cause. It cannot be bacteria that is causing this disease. So he went on further analyzing, studying and trying to find out, and then he chanced upon this particular creature, virus. Now let us study about the characteristics of virus. Virus has both living and non-living characteristics. It's the most important feature of virus. Virus behaves as non-living body outside a host. It needs a host to survive. Virus needs a host to live. Outside the host, it behaves as a non-living thing. It does not show any characteristics of life. It is just as good as non-living thing. So it is considered both as living thing as well as non-living thing. It is in between life and non-life kind of thing. So let us see the living features of virus. They contain nucleic acid. They have either DNA or RNA in their body, which is a living characteristics. Only living things have genetic material. They are infective. They cause disease in plants or animals, even bacteria. Whichever host they will infect, they cause disease in them. Viruses are obligate parasites. They are obligate parasites. They will harm whoever they are in. So they cause infection. So infection is again caused by living things. They propagate in living body. All the action is taken place in the living body only. Genetic material is capable of mutation. Now mutation again is a characteristic of living things. Living things mutate, change in genetic material. It is brought about in living things because they only have the genetic material. So this is a living character of virus. They cause disease. They have enzymes. Virus, they have some kind of enzyme system in their body, which is again a living characteristics. So these are the living features of virus. Let's see the non-living characteristics of virus. They behave as non-living thing outside any host body. They do not grow, respire or show any metabolism. Those things which are characteristics of a living thing, that means we have done the characteristics of living things, isn't it? They show respiration, they do respiration, they show metabolism, they grow, they react to change in the environment, response, stimulus. Those things are all absent in case of virus. They show no response. They cannot grow in artificial medium. Now, all the living microorganisms like bacteria and other things, they all grow in artificial medium. Even plant propagation that is done in artificial medium, they grow quite well. So living things grow quite well in artificial medium. But viruses, they cannot grow in artificial medium. They can be crystallized. This is a very non-living character. Salts are crystallized. So, but virus, they can stay infective after crystallization also. Say they remain crystallized for many years. Again they find a host body, they will again come back to being infectious. So it's like that. So these are the non-living characters of virus. So go through them, living character and non-living character. These are very important because virus being a special kind of entity, these are the things which are associated with it. So you need to know all this. Now let us talk about the structure of virus. Size of virus is 10 nanometer to 350 nanometer. You know how much is 1 nanometer? It is 10 to the power minus 9 meter. Now Do you remember what was the size of bacteria? Bacteria ranged from 0.01 micron to 10 micron, isn't it? That was in micron and this is nanometer. So, can you tell the difference between size of a bacteria and a virus? It will be, virus will be 1,000th of a bacteria. 1,000 times smaller than a bacteria. So, that is their size difference. Now, the shape of virus may be rod-shaped, as in case of tobacco mosaic virus. They may be spheroidal, they may be cuboidal, or they may be dual. Dual means cuboidal and helical. Both parts are there, both features are there. And dual example is bacteriophage, a very good example. Now look here, the bacteriophage. Virus will consist of a protein coat. Okay, so bacteriophage, it also has a protein coat. Now the protein coat here is divided into three parts. There is head, which is cuboidal. Then the body is helical in shape, and then there is tail region. So this whole thing is the capsid. And the capsid is made up of individual subunit that is known as capsomere. In every case, in case of all the virus, capsid is made up of capsomere. And the capsomere are arranged in different shapes. So here it is, the shape is like this. It consists of head, body and tail. The head has genetic material in it. So this is the structure of bacteriophage. Sometimes you are asked to ask question on the structure. So please remember the structure, draw it and practice it. Now let us talk about virion. What a virion is. Sometimes question is asked, what is a virion? Now virion is nothing but a single infective virus. A virus which has found a host and it is about to start its infective cycle, that becomes a virion. So that is it, virion. Virion therefore will consist of genetic material and a capsid. Capsid consists of capsomere, as I have already told you, and sometimes the capsomere may further be covered by an envelope. The capsomere or the capsid and genetic material, they are together known as nucleoprotein, because nucleic material and protein, it is known as nucleoprotein. So it may be covered by an envelope. So that is about the structure of virus. Now let us see the classification. Virus classification. Virus has been divided into two types based on the genetic material present in them. It is DNA virus and RNA virus. Now again, these two types are divided into two subtypes based on whether the genetic material is single-stranded or double-stranded. Both in case of DNA and RNA, it can be single-stranded or double-stranded. That means there are four categories of virus basically. So it will be double-stranded DNA virus or single-stranded DNA virus. Double-stranded DNA virus are poliovirus, herpes virus. Single-stranded DNA virus are bacteriophage. Bacteriophage, they are known as T2, T4 phage particle. They infect bacteria. Most commonly they infect Escherichia coli, the bacteria which stays in intestine. So they are sometimes known as coliphage also. Now RNA virus, again single-stranded and double-stranded. Single-stranded RNA virus example are TMV, tobacco mosaic virus, coronavirus, retrovirus, influenza virus. All these are single-stranded RNA virus. Then there are some double-stranded RNA virus, example reovirus. This is also found in human intestine sometimes, reovirus. So this is the classification of virus. Now we come to a very important aspect of T2 virus, which is phage virus, which is known as lytic and lysogenic cycle. Okay, now let us see what lytic and lysogenic cycle of T2 is. T2 is nothing but bacteriophage virus which infect bacteria, especially E. coli. Now look at the diagram. Here the T2 has landed on the bacteria. T2 particle, one virion has landed on a bacterial cell. what it will do, it will insert its DNA material, which is single-stranded DNA, into bacterial cell. That DNA material will go and unite with the genome of the bacteria. Then what will happen, it will take over the activity of bacteria and it will use it to produce only phage particle. Whatever is required in phage particles, say protein, whatever protein is required, then DNA material, whatever things are required to make phage, the bacterial cell will act as a factory for making phage particle. So ultimately it will make many phage particle, and when the production is completed, bacterial cell will break open, it will die and it will release all the phage particle which will infect further bacteria. So this is lytic cycle where phage is going and killing the bacteria, and in turn it is dividing itself. So this is, then we have lysogenic cycle. Here what happens, a phage has landed on the bacterium and it has inserted its DNA in the bacterial cell. The DNA goes and unites with the genome of the bacteria, but it stays there like that. It becomes prophage. The DNA of the phage is now known as prophage because it has entered the bacterial genome. Bacteria here will not get killed, but it will divide as it does, as the bacteria divides using fragmentation, fission, budding, all those processes it will do, and it will create multiple copies of this prophage. The genome that is here recombined with this phage DNA, when bacteria will divide, it will divide that phage DNA also along with its genome. So here we see a recombination and multiplication of recombined genes with the help of bacteria. So this is lysogenic cycle where bacteria is not killed, but here it is used to multiply the recombined DNA. But here in lytic cycle, the bacteria is killed and the whole bacterial cell is used as a factory to manufacture phage particle. I hope you have understood this. It's very important, lytic cycle and lysogenic cycle of bacteriophage. So I'll tell you once again what happens here in lytic cycle is a phage has landed on the bacterial cell. It has inserted its DNA in the bacterial cell. DNA has gone and joined with the genome of bacteria, and then it has used the bacterial cell as a factory to manufacture phage particle. And ultimately the bacterial cell dies and phage particles are released. In lysogenic cycle, a phage has landed on the bacteria. It has inserted its DNA on the bacterial cell, which goes and unites with the bacterial DNA. But now the phage DNA is turned into prophage, and the bacterial cell remains as it is. It divides or rather multiplies using the usual methods of fission, budding, fragmentation, etc., and produces multiple copies of this prophage. So here we see multiple copies of phage DNA being made, and in lytic cycle we see that the bacterial cell is used as a factory to manufacture many phage particles. So this is lytic cycle and lysogenic cycle. Now we have come to the last slide of virus. Here we are just about to see some of the disease caused by virus and importance of some of the virus in producing enzymes. So let us see disease caused by virus. In case of plants, tobacco mosaic virus, bean mosaic virus, there are mosaic virus seen in many plants. All these are caused by virus. Then leaf curl in tomato, sometimes we see leaves get curled, that is because of virus. In man, pox, influenza, mumps, rabies, measles, polio, all these are caused by virus. Now, enzymes produced by certain virus in biotechnology and other industrial uses, there is a provision for enzymes to carry out different reactions and to carry out different procedure and technique. Now these enzymes are produced from some sources, and virus are a very good source to produce enzymes. So what are the enzymes that we can get from virus? RNAs and reverse transcriptase. These are very important in biotechnology. When we will do that, then you will come to know the use of these enzymes. As for now, just know that they are procured from retrovirus. Then protein kinase virus comes from herpes and adenoviruses. DNA dependent RNA polymerase comes from pox virus, and lysozyme is produced from phage, T2 phage. So these are some of the enzymes produced. Now just one last thing is left, let us discuss that also. What are virions? These are small viruses which has ssRNA, single-stranded RNA as genetic material, and they infect plants. So this is what virioid is. I hope you have understood this topic virus. Please go through the slide again and again so you can be clear about all the concepts.